Hello, everyone. It's a pleasure to be here. I want to thank Martin for the invitation and for the opportunity to speak here about my research. I'm going to be talking about reproductive aging and the effects from the NAD metabolome on the ovary. So starting off a little bit about my background, I work at VitaDAO currently, which is an alternative funding model for aging research. Before that, I was in traditional VC and venture creation for a bit, looking at commercial opportunities in aging and reproductive aging. But for this talk, the most relevant affiliation would be the one with University of New South Wales, where I did my PhD. So Miguel has introduced a lot of these concepts, so I'm gonna go briefly over the introduction, but I think it's a good overview of why I think reproductive aging is something that's very important to study and fund. So we know that ovarian function declines with age, and it does so rapidly and before everything else in the body. Fertility is a very good functional marker of that. However, ovarian aging impacts much more than fertility and is directly related to female health span. This graph is just an example of one condition. It represents cardiovascular disease onset risk. And as you can see, before menopause, women are at a much lower risk, but during the menopausal transition, they catch up and exceed men in risk, leading to that sustained higher risk after menopause. And beyond health span, we might make a case that ovarian age is also linked to lifespan because we see young ovary transplantation into old mice significantly increases their lifespan. And speaking of lifespan, our lifespans, or more precisely life expectancy, has been increasing over the past century. However, the age of menopause hasn't budged at all. Which means that nowadays women spend 40% of their lives post-menopause, which comes with increased risk of this fine selection of age-related diseases that comes along with it. And it also means that women spend around 34% of their life in poor health, in comparison to men, for them is only 26%. We know that late menopause also correlates with extended longevity. For every 1 year of later menopause, there is 2% reduction of mortality risk. We know that women who go into puberty later, into menopause later, they have higher chance of living up to 90 and over. And of course, there is a big genetic component to it. We also see brothers of women with later menopause live longer. And there was a very interesting human genomics study that linked some DNA repair pathways to later menopause, which one of the panelists, Dasha Stankovic, I hope is going to cover a bit later. So what is actually ovarian aging? I was told it's illegal to present here without showing a graph of the hallmarks, so I found a paper that has come up with hallmarks of ovarian aging. They're pretty much the same, but they seem to manifest earlier in the ovary, especially markers of inflammation and senescence. But the other side of it is ovary-specific. As Miguel mentioned, a woman is born with all of the eggs she's ever going to have, and there is a steady decline over time and a countdown to menopause. So actually the biggest or the largest quantity of oocytes is in the human female fetus, around 7 million, which majority of them are lost, and at birth you have around 1 million, which steadily decline until that feared age of 35 when your ovaries are termed geriatric. And after that, there is a much steeper decline and accelerated follicle loss. And we still have no idea why that happens. On the other hand, you have oocyte quality. And this graph here represents the effect of oocyte quality on fertility and live birth rates. So we see in blue, those are maternal oocytes, so oocytes collected from the patient, and IVF efficacy declines with age. However, if oocytes from a young donor are used, there is no change in pregnancy rates with age, which until 44 years, which is shown here, which means that the female reproductive system is perfectly capable of carrying out that pregnancy at that age. So I'm not gonna go into much detail here, but the ovary has a very dynamic architecture, and it's constantly changing throughout the cycle. But what I want to highlight is the small follicles on the left. They're called primordial follicles, or the ovarian reserve, and they stay dormant for most of a woman's life, and every month after puberty, a bunch of them are activated each month, around 1,000, but only one ovulates. So what are some strategies to target ovarian aging? Similar to aging, we have the slow down approach and the reverse approach. For slow down, we have some geroprotectors or aging interventions that might also impact the ovary, or ways to reduce follicle activation rate or get less of those primordial follicles activated. On the reverse side, we have tissue cryopreservation and in vitro gametogenesis. So both of those would then have to be reimplanted back into the patients. And that sounds a little bit more sci-fi, but tissue cryopreservation is actually in clinical practice now, but not for reproductive aging, but rather for cancer survivors who have undergone chemotherapy and they are too young to freeze oocytes or embryos. So that's their only option. So I'm going to focus on the aging interventions today. And before I go into NAD and my research, I want to go through a crowd's favorite, which is rapamycin, because rapamycin has shown some of the best results. It's also shown effective in chemotherapy patients, and is currently going into human clinical trials for reproductive aging. And while many geroprotectors have been tested and show some positive effects, from senolytics to anti-inflammatory and antioxidants, rapamycin not only addresses the aging side, but mTOR is also directly involved in oocyte activation, so dormant follicles becoming growing follicles. And now going into NAD, this was a paper published by my PhD lab a couple of years back, and it was led by Lindsay Wu, Robert Gilchrist, and David Sinclair, and Hayden Homer. So I'm going to briefly summarize the results from there, which basically showed that NAD repletion restores fertility in reproductively aged mice. I'm pretty sure most of you know what NAD is, but very briefly, it's an essential cofactor for redox reaction, but also for non-redox NAD-dependent enzymes, from sirtuins to PARPs, etc. And it's directly and indirectly involved in many key cellular processes, from metabolic pathways to DNA repair, to mitochondrial function, etc. So maybe the biggest finding was that NAD supplementation in advanced maternal age in mice restored fertility, and it did so almost improved the birth rates by almost more than twice. It also increased litter size and pups per mouse. But there is one important caveat, which is it only did so at the lower dose that we administered, and the effect was lost when the dose was increased. Oocyte yield was also improved in the both mouse strains we tested, which was Black 6 and albino, Swiss albino. So of course we had to check if NAD is actually going down in the ovary and in the oocyte, and if our NMN supplementation boosts NAD. And we see that there is this oocyte-specific NAD decline. So NAD does decline in aged oocytes, and it is boosted back up by NMN supplementation. However, we do not see that in the whole ovarian tissue. The NAD effect on the oocyte links to the results we see in oocyte quality. The NMN supplementation improved dramatically spindle assembly, which makes sense because we gave it 4 weeks before we collected the oocytes, and the M1 mitotic division, which segregates the chromosomes, occurs 2 to 3 weeks before ovulation, and that's the most ATP consumption intensive stage of the process. SIRT2, which is an NAD-dependent enzyme, also is involved in kinetochore microtubule attachment, and deacetylase BABR1, which is a key enzyme in that process. Just a quick note, it does also work in vitro. So if the mice are untreated but NMN is supplemented in the oocyte culture, it also seemed to improve oocyte quality. And while that's irrelevant for reproductive aging, it is quite important for IVF, and the need and demand for IVF has been increasing over the years. Lastly, we looked at some transgenic models, and what we see is that SIRT2 is sufficient but not required for improvement of oocyte qualities at later ages. So what does that mean? We overexpressed SIRT2, and we see improved oocyte yield, oocyte quality, spindle assembly, and pregnancy rates. So it is sufficient to do that at later age without NMN supplementation. However, when we look at a SIRT2 knockout model at younger ages, it is not needed. So those mice still have perfectly healthy spindles at younger age when NAD is still at a high level. We also looked at 2 other transgenic models, which are for NMN-AT1 and NMN-AT3, which are NAD synthesis proteins. And they have different subcellular localization. So NMN-AT1 is localized to the nucleus, NMN-AT3 is localized to the mitochondria. And what we see is that there is oocyte yield rescue only in NMN-AT1, which means that NAD subcellular localization might also play a role. So luckily, after this study, there are a number of other ones that came out from different labs that supported our findings and built upon them. For example, another study shows, again, that quality of maternally aged oocytes is increased. There is also one that addresses the number of follicles rather than the quality, and shows that long-term NMN administration seems to positively impact the diminished ovarian reserve with advanced maternal age. And very recently, there was a study in rats that also showed improvement in ovarian aging with NMN. And the mechanism behind that was the balancing of mitochondrial fusion-fission reactions. Now I'm going to shift gears a little bit and talk about the main project of my PhD, which is still unpublished work and a bit more new. So I talked about the decline in follicle number with aging and the acceleration of follicle loss after 35. However, if you looked at the effect of chemo, this happens much more dramatically and much faster in chemotherapy-treated cancer survivors, which leads to exhaustion of the follicular reserve and earlier menopause. How that works is chemo is quite toxic to the ovary. It obviously targets very rapidly dividing cells. So that means that it kills the growing follicles. But these follicles are quite important in keeping the ovarian reserve dormant. So they send signals that prevent dormant follicles from activating. And when the growing follicles are gone, there is this feedback loop of a lot more follicles starting to grow. They're getting killed by the chemo, and that results in exhaustion of the follicular reserve. So we looked at whether the chemotherapy treatments that we choose to use indeed diminished fertility in mice. So we used doxorubicin and cisplatin. Of course, they both dramatically reduced fertility in mice, and NMN supplementation recovered that. And here, I'm showing you two experimental setups that we used. One of them is co-administration of the chemotherapy and NMN, which means that there was a single chemotherapy injection given together with NMN, which is the DOX model. And for cisplatin, we gave cisplatin first, then we had a wash-off period of 2 weeks, and then administered the NMN, which is given in drinking water for a sustained period of time. So this is important from a mechanistic perspective of how NMN might potentially work, but also it might be relevant in clinical practice because sometimes chemotherapy can be delayed until you find something else to mitigate its negative effects. And we also see that in oocyte yield is decreased with chemotherapy, and it's boosted back up after NMN. So, of course, for this study, we made some assumptions that NAD declines after chemotherapy, and we thought that's the case because the chemo causes DNA damage, and that activates the PARP family of enzymes, which act on the DNA repair front. They use NAD, and it declines. So indeed, that's what we see. The chemo-treated groups have lower NAD, and that's boosted back up after our NMN treatment. The other very important thing to check was whether there is any impact or negative impact of NMN on the efficacy of chemotherapy or on tumor volume. And there was no negative effect. Actually, there is a additive benefit from NMN when supplied with a chemotherapy treatment. And in certain cases, NMN decreased tumor volume more than the chemotherapy that we administered. The graphs that are shown here are actually the same study performed in 2 independent labs to validate the result. We looked at another cohort of chemotherapy-treated mice, assessed fertility. Again, we have the same result. This is cisplatin co-administration, but again, fertility is lost with cisplatin and recovered with NMN. However, we also wanted to see if there are any effects beyond the ovary and looked into late life health of these females. So one key concern after menopause is osteoporosis, so I decided to have a look at the bones. So I don't know how well you can see the effect here, and if you can tell which one is the chemotherapy-treated one, but we saw a very big effect in bone density is the bottom left, that is chemotherapy-treated, and next to it is the chemo plus NMN, which seems to have recovered density. But this is quite rare to have such a big impact on cortical bone. So we did quite a robust analysis. We assessed 300 sections per bone for the tibia and 400 sections of the cortical bone for the femur and 100 sections for the trabecular bone. which is quite extensive. It was semi-manual, a very lengthy process, and probably partly why I don't do research anymore. But we did see some pretty exciting results. The tibial cortical density, thickness, and volume were rescued with NMN after chemotherapy. We see the same thing in the femur. And we also tested tibial physical strength. So we did a 3-point bending mechanical testing, which is basically the bone is rested onto surfaces, and there is a lever coming on top, which measures the force that's required to break it. And we see a significantly increased bone strength in the chemo-NMN group when compared to chemotherapy in the tibia, and a similar pattern for the femur that did not reach statistical significance. So here maybe it's relevant to mention that maybe you see there is different number of mouse per group, and those mice were quite old. They were 26 months, just over 2 years, and some of them have died before the collection date, which is why we have less bones in certain groups. And notably, the highest number of animals was in the NMN untreated group, so no chemotherapy and NMN, and the least animals were obviously in the chemotherapy group. So I'm not going to have much time to tell you about the finer details of that study, and I'll be flattering myself if I say I'm leaving you on a cliffhanger, but it's quite an interesting story. If you're thinking it's estrogen related, that's what we thought as well, but it's not. It's a different mechanism. But maybe stay tuned for our next paper to find out. I think I want to talk briefly about safety, because I always get questions from women on whether they should take NMN or not, and which NMN they should take. And I want to go back to the finding that we have on NMN dosing. So NMN increased fertility, pregnancy rates, pups per mouse, oocyte yield, etc. At the reproductively aged mice. However, it only did that in the lower dose, which is 0.5 grams per liter. And the higher dose of 2 grams per liter was shown to be not effective for the same measurements. We don't really know what's happening there, but we have a few suggestions. There is an upper limit to NMN tolerability. There is an optimal range for dosing. There could be potentially accumulation of NMN degradation product, niacinamide, that could be an inhibitor for SIRT6 or other sirtuins. Or the higher dose could be beneficial for oocyte quality, but it might have adverse effects on other aspects of fertility, so the cumulative effect is no effect. And as NAD supplements are commercially available, there is an important concern there on how people are using it. Of course, another important concern to address when we're talking about pregnancy and fertility is the offspring health, and we did test that as well. There is no change on offsprings after maternal NMN administration. There is a little bit of an increase in lean body mass, which is arguably good. And then there is another study that suggests NR, or nicotinamide riboside supplementation during breastfeeding. It is not harmful and it might be beneficial for offspring health. So to summarize, we used pharmacological and genetic approaches, and both of those showed that NAD enhances ovulation rate, oocyte quality, pregnancy rates, and pup number or litter size in aged female mice. We also see chemotherapy protection, so NAD repletion or boost by NMN supplementation. increased fertility during chemotherapy, increased fertility after chemotherapy, and also had impacts on late life health, specifically bone health, as it increased volume, density, and strength of the long bones. And lastly, a word of caution for the optimum dose for NMN. And there might be some adverse effects that we not know about, so we do not recommend anyone taking it without further study that elucidate the optimal dose. And with that, thank you, and happy to take questions.
We have questions in the audience.
Thank you for your talk, for your great talk. No, and you don't recommend NMN, and I understand that. No, but I'm following the advice. Dr. Sinclair doesn't give advice, but he takes 1 gram of NMN a day. I also took it for years. No, but what do you think about the? Can you say something about the dose for humans at the moment?
No, I think I'll leave that to medical doctors. I don't want to give any medical advice. I'm not saying I'm not recommending people take NMN. I'm not recommending anything. It's just I think there are some unknowns in the space, and it might be something that's particular to the ovary and the follicles, so it might not affect you. There might be other effects we don't know. It's
okay, thank you.
Maria, really interesting talk. But looking at some of the graphs you showed, it's always struck me, what's so special about 35? Why after 35 you have such a steep decline?
That's a great question. We have no idea. I think that will be very interesting to answer here.
Thank you, Maria. It was great. I have a question about the mechanism involved to protect the ovarian reserve. And do you know if it's affecting P63 or CHEK1 or CHEK2 as a potential way to inhibit the apoptosis in those cells?
So for the ovarian reserve, I mentioned one paper that came out after ours. However, I also counted a lot of follicles for our studies, and I did not see a change in the primordial follicle number or any other follicle type. We did see some change in follicle health after chemotherapy, so NMN improved follicle health, but not follicle number. So from what I see in our studies is oocyte quality and maybe improved ovulation rate, but